Yeah, so can you share my see my screen?
Yes, you can go for full screen now.
Yeah, I'll go ahead. Yeah. Right.
Okay, right. So before that, just to give some introduction, then we can start.
Sure.
So good evening to all the participants. I welcome you all for our evening guest lecture. I thank our EEE department and our principal and vice principal sir for giving me this opportunity to organize this guest lecture on recent advancements in biosensors, and which will be handled by Dr. Fidel Kumar, project officer, Center for NEMS and Nanophotonics, and as well as he is an entrepreneur also. Fidel Kumar is separately, he's having as a managing director, he is running an industry termed as JSP Enviro Private Limited at Chennai, but he's an officially he's a project officer at IIT Madras, and he did his PhD in biotechnology in IIT Madras also. And I welcome our chief guest for this expert persons, Dr. V. Fidel Kumar, and hand over the session to him. Sir, welcome you.
Sure, thank you. Thank you for this such a nice introduction.
Yeah.
I'll be talking about some advances in the biosensor. So how I have divided this topic is I'll be going over some of the components or parts of the biosensor, how it is organized, and then I'll be giving some examples like what are the advances and where all it is used. So I hope this talk will be a bit more informative for everyone who is attending this.
Yes.
Yeah. So basically what is sensor is it measures a throughput where if it constantly measures some kind of an analyte or something that is present in the solution or ambient air or something like that. So it constantly measures something. So where do you use it? I have just put a small pun. So we use it to measure whether our employees are really working or not. So we will be constantly monitoring whether they are really, giving their 100% or not. So just as a jokes apart, so what I'll be doing is I'll be going through the different aspects of the biosensors, and then I'll be giving a real-time application like where all it is used. So how the biosensor is different from the other normal sensors is, so there are actually 5 parts in the biosensor. So I'll be trying to highlight it all over here. So first thing is, what are you trying to measure? So what kind of a bioanalyte are we looking to measure? For example, it can be something like glucose, or something from the blood, or it can be something like alcohol in the breath or something like that. So what are we trying to measure? So that is a bioanalyte. Depending upon what we want to measure, there are different recognition elements. So in our body, each of this component is recognized by a particular set of elements that are present in the body. Now this is what we call as the biorecognition element. Okay. So for example, glucose will be recognized by some specific molecule. So that is the biorecognition molecule. Now what we do in biosensor is we put this biorecognition molecule on a base matrix where we can immobilize it on some base matrix, and on the reaction with the analyte, it produces some kind of a signal, and this signal is passed on to a special part called as transducer, okay, which analyzes the signal. And finally there's an algorithm to tell like how much of this bioanalyte is present. For example, again going to back to the example of the glucose, it will tell what is the concentration of the glucose in the blood. So moving on, so there are 3 ways the transducer can pick up the signal. Okay, so the few of the common ways that is used in the industry is either one is optical, so where there is some chemical reaction happening and it produces some kind of a color, and this color is picked up by the transducer element, which gives out as a measurable output. So that is usually classified as the optical transducers. Other kind of transducers are electrochemical, where I'll be talking about where each of this fit with the biorecognition element. But in electrochemical, so the beauty about most of the processes that is happening inside the body is it happens at a certain potential. Okay, so some of the reaction happen at a certain potential. So what electrochemical transducer do is it constantly applies this potential, and it measures the current that is produced after the reaction happens. So this particular reaction happens. And the third kind of transducers that is very commonly used is mechanical, whereby the actual change in the mass is measured. Okay, for example, if there is some bioanalyte coming and binding to the surface of the transducer, it changes the measure in the mass. So there are basically three kinds of transducers that is used in the market. Okay. So I'll talk a bit more about the biorecognition element over here. So what exactly is biorecognition element? So if you consider our normal biomolecules that is present in the body, so these biomolecules, there are three kinds. Okay, so the three of the major biomolecules, one is protein, okay, which is made up of amino acids, and the second one is DNA, which is made up of nucleic acid, and the third one is lipids. Okay, so two of the major biorecognition molecules that is used is proteins and DNA. Okay, now proteins, the way they recognize the molecule, there are two kinds. Okay, so it can either undergo a catalytic reaction, by which it react with the component. For example, it can react, if there is a biorecognition molecule for glucose, it will react with glucose and it will produce some electrons. Okay, now these electrons can be measured electrochemically by transducer. Okay, the second way of doing it is based on color. Okay, so it reacts with the analyte and it produces certain kind of color. Okay, and this color can be measured optically. Another way of going about is actually the change in mass, which we'll be covering in a bit more detail. This is what you might be commonly known as antigen-antibody interaction. Okay, so what we do over here is we immobilize a special protein called as antibody, and we detect antigen. Antigen is the protein that is produced either by the pathogens or in case of some infection, they release some toxins. These are all certain kind of proteins called as antigens. So we detect this using the mass. The next most popular is nucleic acid. So just to give a gist, everyone might know that DNA is a sequence of code which is made up of A, T, G, C. So what we do in nucleic acid biorecognition molecule is we try to detect the DNA from the pathogen or something. So we immobilize the complementary DNA to that pathogen on the surface of the transducer. And whenever there is a pathogenic DNA or the DNA from pathogen which is coming, it will come and bind to this DNA and it will produce either the change in mass or it produces change in something called as impedance, which I am pretty sure you guys might be aware of. Okay. Lipids are not usually used because they don't have that kind of a capability of recognizing some other molecule. So mainly what we do is majority of the biosensor uses proteins, and proteins will be one of this type: catalytic, color, or mass. Whereas DNA is used in certain cases. So these days, with the advancement of the COVID, most of the sensors that we use is actually DNA-based sensors, which we commonly call as PCR. Okay, so I'll be talking about that also in great details in the later thing. So these are the three kinds of biorecognition molecules that is usually used in sensors. Okay, so moving on. I'll be talking about certain examples of the sensors, like how it is used. So one of the common sensors that you might be seeing in the real life is the breath analyzer, which is actually used to detect the alcohol in the breath. So this breath analyzer uses transducer most commonly as the electrochemical transducer, which I was earlier talking about. So the basic mechanism over here is once you blow the air to this sensor, the alcohol in the breath is broken down into acetic acid. And acetic acid, this process of breaking it down into acetic acid requires oxygen. So basically, the reaction that is happening over here is that the oxygen reacts with hydrogen ion and 4 electrons to give water. Now, there are two components over here. There is a cathode and an anode. So basically, this reaction happens at the cathode. Now, at the cathode, there will be a dip in the current, which is the 4 electrons that is taken during the reaction of conversion of ethanol to water. Now this electron that is taken up is recorded as a current. Okay. Now in the sensor, usually this calibration might be done where people might have already set a calibration value with concentration of the alcohol and current, right? So when the transducer gives the current, it plots, actually the algorithm plots the current of the unknown value, and it finds out the concentration of the alcohol, which is actually given out as the output reading. Okay. So this is one of the common examples that is used in. These days, there are other, this is not exactly a biosensor because it directly converts the alcohol into the required current. But there is a biosensor thing that is also developed for this, which is conversion of the alcohol using an enzyme called as alcohol dehydrogenase. Okay. So what this alcohol dehydrogenase does is it reduces the alcohol, and there is a component called as NADH, which is converted to NAD, and it produces electrons. And this electron is measured as an output signal, okay, which indirectly gives the concentration of the alcohol that is usually present in the blood. So this is one of the common examples of the electrochemical sensor that is available in the market and very highly applied in the market. Now coming to the next one, in the airport, very commonly they check out for the presence of the explosives and the drugs. So how they usually do is they have like a white cloth. They wipe it if a suspicious person is there. What they do is they take this cloth and they wipe it in the bags and on the surfaces, and they subject this wiped material to the electrochemical sensor. Okay, so most commonly, if there are certain drugs, so what they do is, so in this case, what they do is they, for example, if they are looking for a drug for like methamphetamine, what they do is they put an enzyme which can oxidize this methamphetamine. Okay. And on oxidation, usually in chemical reaction, it always produces electrons. Okay. Now this electrons is measured as an output signal, same as the earlier, and it gives the concentration if there is present any. Now this can't be done if you have a bulky equipment. So the most common way of doing this is to miniaturize the whole equipment. So the way to miniaturize the equipment is by something commonly called as the 3-electrode system, which is used in very commonly used in the electrochemical setup. So for example, what you see upper part over here is that there are 3 electrodes. So one of them is called as the working electrode, on which the enzyme is attached. Okay. The second, the center part is the working electrode. The second part is counter electrode. So whenever an electron is produced from the working electrode, it travels and it goes to the counter electrode. Okay, thus completing the circuit. Now there's a third electrode they use called as the reference electrode. Now this reference electrode is very essential because, as I said, most of the biological reaction happens at a particular potential. So for example, so NADH is converted to NAD plus electrons at a potential of minus 0.32 volts. If you go beyond this, if you go lower than this potential, or if you go higher than this potential, this reaction may not happen, or it may not happen at a very high rate. So this reference electrode, the job of this reference electrode is to maintain a particular voltage between the working and the reference electrode. Now this can be, so, and the job of the counter electrode is to complete the circuit. So this kind of system is called as the 3-electrode system that is very commonly used in sensor, electrochemical sensor. So because of this 3-electrode system, it produces a certain amount of current, and this current is recorded by transducer that is attached along with the sensor. Okay. Now job of the transducer is done once it gives the value of the current. Now the next is done by the signal output, which has a calibration on which this value of the current is plotted, and it gives you the concentration of the analyte. Okay. So this is a very quick method since it's electrochemical, and enzyme also acts in a very short span of time. So it's a very quick method to actually detect if there is any component that is present over there, which was of suspicious nature. Now this is slightly different. This is very commonly used for drugs, but it's not so much commonly used for such as explosives. For explosives, they use a sophisticated technique called as mass spectrophotometry, because explosive can be of different nature. So you can't have enzymes for each explosive. But since most of the drugs come from either the synthetic nature, you can have enzymes that can be synthesized in the lab as a biorecognition element. But this can't be done for explosives. So this is one of the most commonly used sensor in again in the security sections. The next common type of sensor is optical sensor, and the very common thing that is used is in DNA hybridization. Now, how do we use it in DNA hybridization? DNA is very conserved for each organism. So there is a special section of DNA called as 16S DNA. So 16S DNA is a special short span of sequences like ATGC, around 1,500 sequences, which is actually conserved for each organism. So just to give you a short hint, so if say usually how the DNA base pairing occurs is that A is bonded to T, G is bonded to C. So whenever DNA usually has a double strand, if there is a on one strand, there will be T on the other strand. If there's a G on one strand, there will be a C on the other strand. So this sequence are like complementary to each other. So say if you want to detect a particular organism, all you need to do is you need to take the transducer and immobilize the complementary strand. So for example, I'm planning to detect say X organism. I'll take the 16S DNA, which is usually present in the library, 16S DNA of that particular organism, and I'll immobilize the complementary strand on the surface of the transducer. Okay. Now what I'll do is I'll take the sample from the clinic. Okay, it might be sputum sample or blood. I'll extract this 16S DNA from the sample and I'll drop it over here. Okay. Now this is done in such a way that when it binds to this particular sample, it has a fluorescent molecule on the top. Okay, only if it binds completely, it gives a signal. Okay, optical signal. Now this optical signal will look something like this over here, which is shown on the right hand side. Okay, so only if there is an organism present in the sample, it will flash out fluorescence or light. Now this is very commonly used in a technique called as ELISA. Okay, which is used to detect various kind of diseases. So whenever the organism is present, there will be a hybridization that is happening, and it is not only used to identify the organism, but it's also used to identify different kind of genetic disorders. Now what I've shown over here is the live image of how it looks like. So whenever you can see that whenever there is a hybridization happening, there is like a red signal given out, which indicates that the organism of the interest is present. Okay, so this is called as DNA hybridization. Now, very commonly, you might have heard of RT-PCR test, which is commonly used for the detection of COVID. So what happens in RT-PCR test is you have a short stretch of DNA, which is complementary to the COVID. When it binds to the COVID DNA, you first amplify the COVID DNA, and when if it is present and when it binds to the COVID DNA, it gives out a signal whenever there is a binding. Okay, and this is very sensitive. If there is no binding, if the signal is below the threshold, it's considered as sample is considered as negative. But if the signal is above the threshold, it's considered as positive. So based on how much the signal is produced, you can really see a live stream of whether the organism is present in the sample or not. Okay, so this is very commonly used. Now, another way of doing this is you can, instead of using optical sensor, you can also use mass sensor over here. Now, since whenever a second strand of DNA is coming and binding over here, it's causing an additional increase in the mass to the transducer. So if you have a mass transducer attached over here, what it will detect is the increase in the mass, which I'll be talking in the next slide, like how do we use the mass transducer to actually detect the samples. Okay. So moving on. So this is what one of the sensors that we are actually working in the lab. It's called as cantilever sensor. So cantilevers are like a thin beam of resonators. What it has is it has a particular thickness, and it is like usually self-standing. Okay. And whenever there is a mass that is attached over here, the thickness is such that it causes it to bend. Okay. Now, there are two ways of using this cantilever sensor. One is static mode and other is dynamic mode. Okay. In static mode, what we do is whenever there is an attachment of mass, it usually deflects from the center point. We usually measure the angle of deflection. Okay. So higher the mass, higher will be the angle of deflection. Okay, so that is how you use the static mode. But sometimes what happens is that in static mode, the problem is if during the production of the cantilever itself, if it's under high stress, it will be already deflected, and you might not get a very sensitive reading. So most of the people usually they prefer the dynamic mode. So what happens in dynamic mode is you also you have the cantilever, you give a ray of light on it, the laser light focused on the tip, because of which it constantly keeps vibrating at a certain frequency. Okay, and this frequency of the vibration is directly proportional to inversely proportional to the mass. Okay, so if there is a higher mass attached on the surface, the frequency of the vibration at which it is fluctuating, it might decrease. So that is what you are seeing somewhere over here, where you can see that the cantilever is vibrating based on the laser beam that is focused on it. Now, one way of how we used this as a sensor, and this was done in our lab, is so what we did is we immobilized a special kind of protein called as antibody on the surface. Okay. So antibody are like special kind of proteins that are produced in defense. What I mean by defense is whenever there is a pathogen that is entering in our body, so our body responds by producing a complementary protein to trap that pathogen such that it won't move somewhere else. Okay, so these proteins are called as antibody. So usually what we do is this defense mechanism is similar even in organisms like mouse and goat. So antibodies for the same pathogen can be also produced in organisms like such as mouse and goat. So what we usually do is we produce the same antibody in the mouse and we immobilize it on the surface. So because of this immobilization, say if you have a blood sample and if you drop it on the surface of this cantilever, so if the same organism is present in the blood, it will be trapped because of the antibody. It will be held onto the surface by the antibody. Now this obviously causes the change in the mass on the surface. Okay. Now, as I said, this is a dynamic mode of response. So the cantilever will be continuously vibrating. And because of the attachment of the mass, there will be change in the frequency, which is usually the decrease in the frequency. Okay. Now, what we did is we already had a calibration of the attached mass with respect to the frequency. So we know that the frequency continuously decreases with the attached mass. So once you have this calibration, when you put an unknown sample, you'll get a change in the frequency. You can plot this change in the frequency and you can find out the attached mass on the surface. Okay. Now, this can be used, this change in the mass is very sensitive because the cantilevers that we actually fabricated in our lab was of thickness around 165 nanometer. So they had a very high resonant, very sensitivity to its resonant frequency. So even if there is a change in the mass, according to in our lab, the measurement showed that it is sensitive up to, and it can detect up to a mass of around 415 femtograms, which is really small. So we were able to detect an antigen at the level of, with the limit of detection of 415 femtograms. Another thing we did was use it for the detection of analytes like urea. So here we did a certain modification. So what we did is we used this cantilever and we deposited a thin layer of aluminum metal on the surface. Now, what we did is, once the aluminum was deposited, we immobilized an enzyme called as urease on the surface. Okay. Now, again, the same method. There is a laser, and laser is continuously causing a deflection, which is giving a frequency. Now, once the urea is put on the surface, say if you have a sample containing urea, what urease does is it converts this urea to two components. One is ammonia and carbon dioxide. Okay. Now, ammonia is known to be an etchant for aluminum. So what it does is it converts aluminum to aluminum hydroxide. On this conversion, what will be happening is, if there's a high concentration of the urea, you will be losing some amount of mass as aluminum hydroxide. So the weight on the cantilever will be actually decreasing. Okay. So here we got a reverse response. So with the increased urea concentration, what we found was the resonance frequency was increasing. Because as higher the concentration of the urea, more will be the aluminum that will be etched out from the surface, and the resonant frequency will be obviously increasing with the time. So we had a calibration curve, and we saw that we could detect actually the aluminum in the range of nanomolar concentration, which is like really low. So in nanomolar concentration means it will be in the range of few nanograms of urea. So this kind of mass sensors is used especially for detecting the mass which is at a very small level, which is around 10⁻⁹ to 10⁻⁵ grams of the mass. So it's considered to be a very highly sensitive sensors. So yeah. So here is one of the, just wanted to show how the dynamic mode works. Whenever there's attachment of mass, it just measures the resonance change in frequency. The third kind of sensor, that is another kind of sensor that is very commonly used instead of current or optical or mass-based, is electrochemical impedance-based sensor. So not all molecules can react and produce color, whereas not all molecules can react and produce electrons. There are certain molecules which can only bind, it won't undergo any kind of reaction. But since the advantage is most of the biomolecules are non-conductive by nature. So what we do is we do something called as electrochemical impedance spectroscopy. So what we do in electrochemical impedance spectroscopy is we again immobilize the biomolecule on the surface, the biorecognition molecule on the surface. Now, by nature, this biorecognition molecule is, as I said, non-conductive in nature. Okay. So because of this, the solution, it offers a certain kind of solution in this resistance in this particular layer where it is immobilized. Now, this resistance can be measured using electrochemical impedance, which gives like a two-part. It gives actual resistance and the imaginary part of the impedance. And usually what we do is we measure these radius for actually measuring what is the resistance it is offering on the surface. Okay. Now, whenever this molecule is already non-conductive, whenever it binds to antigen, what it is doing is it is adding another layer of resistance on the surface. So this radius, the resistance radius that is actually offered to the solution onto the transducer, is actually increases. So below you can see a study where just antigen or antibody, it is showing a certain kind of resistance, whereas when you add antigen, which is also a non-conductive part, you can see that this resistance increases. Okay. Now this resistance can be measured as we call it as the charge transfer resistance. That is, it is the resistance offered by the solution or the immobilized layer for the transferring the charge onto the underneath impedance transducer layer. So this charge transfer resistance can be measured and calibrated, and you can have a sensor which is called as impedance sensor, which measures actually the molecule based on how much the resistance it offers onto the surface. Okay, yeah. So this is not very commonly used sensor, but it is also one kind of thing that is coming up this is one alternative that is coming up. So based on this was some of the examples that I had given, like on the different kind of sensors that exist in the market. Now, whenever it comes to biosensor, it's a compromise between two. So either it can be a real-time sensor where it is continuously sensing some molecule and it is giving you, an output. Like for example, continuous blood sensor. If you implant it on, you can have something like a skin patch on the surface where it continuously tells you some, component. So this is very commonly used in sweat sensors where it tells you what kind of ions is present in the body. Okay, so the function of this sensor is real time. It continuously gives you some parameter, or it can be used for the detection of diagnosis, which we use commonly for the detection of the COVID, where once you put the sample, it continuously tells you how much of the infection is present in the body. Or it can be like for the blood analytes like glucose or something. The other kind of sensor is ultrasensitive, where there are two parts. So ultrasensitive is used where you have a very dilute solution. For example, contamination in a lake. So lake has a very huge volume of water, right? So whenever there is a contamination that is happening in such a huge volume of water, the contaminant will be, there's a chances that it will be diluted to a very high extent. So in that case, you need something called as ultra-sensitive sensor, okay? So which can detect something in very minute quantity. So this quantity is usually, given as ppm or ppb, which is like parts per million or parts per billion, which you might really commonly hear in terms of air quality measurement, because the contaminants are diluted into it. Or it can be something like onset of a disease where the amount of pathogen might be very less in quantity. So in this case, you need a very ultra sensitive sensor. But most of the biosensor, it's usually a compromise between real-time sensor and ultra sensitive sensor. So depending upon what kind of a nature you are looking for, it will be a compromise between these two. So I think I just try to keep this presentation, sweet and short, so that just giving you a gist of what are the different components of the biosensors and how it is used in day-to-day life. I think my thing is done over here, and I'm open for questions right now.
Thank you, Fidel Kumar.
Yeah.
So this area is totally a different area for us.
Right.
We have deal with sensors, electrically based or electronic based. This one is totally different. Biotechnology or biosensors is totally a new area for us. It's very interesting. And as well as I want to have a question. You have mentioned as cantilever based some sensors. Where these find applications? What could be the, whether you have tested the accuracy of those systems?
Yeah. So we have tested with some of the clinical samples also the accuracy of this cantilever sensor. It's more or less in the range of plus minus 2% or 3% compared to the clinical test. So one of the places where cantilever sensors can be really used is like other than the samples of the blood. So blood is very commonly used as the sample for the detection of some kind of analyte. But other than blood, there is like cerebrospinal fluid or there is lumbar fluid, where there are certain rare disorders where the protein molecules produces in very dilute concentration. So in this case, cantilever sensor acts as a very good thing because of its high sensitivity. Most of the frequency-based sensors have a very high sensitivity. That's why it's termed as ultra-sensitive. So other than the blood analyte, if you are looking for something like disorders in other fluid, detection in other fluid, cantilever sensor can be a very place of work.
Okay, right. Yeah. So any other queries from participants, audience can have a discussion with the expert persons. Any doubts we can clarify with him. Right. So if there is no doubts, means we can close our sessions. Sir, HOD sir, hello sir. Ilango sir.
Yeah.
Great. Okay, right. So, okay, thank you, Fidel sir, for your valuable.
Thank you for inviting me for this talk. Yeah.
Thank you very much. So,
Yeah.
for the participants and audience, we have posted the feedback link, and you can fill the feedback link. Thereby, you can receive the e-certificate for that. Thank you, all the participants, as well as thank you very, very much, Fidel sir.
Thank
For
you.
valuable
Thank you, Karthik.
insights.
Yes.
Yes, thank you. Thank you very much. Thank you all.
Bye.